The next speaker is Emanuela Fabiola Craparo. She has discussed her PhD on new polymeric networks as drug delivery system, and her research was focused on synthesis and physiological characterization of new hydrogel obtained by UV radiation. Now she spends a training period in Praga. Now she is a researcher at the University of Palermo, and the research field is on chemical synthesis, chemical, physical, and technological characterization, bioevaluation of new polymeric and lipid systems for modified release. The title of her presentation is Lipid and Polymeric Nanostructures for Brain Drug Delivery and Targeting. Thank you.
Thank you for your presentation. Good morning to everybody. The topic of this presentation will be the description of colloidal nanostructures, such as solid lipid nanoparticles and polymeric micelles, as potential carrier for drug delivery and targeting to the brain. In fact, after in vivo administration, the drug efficacy is affected by many factors, such as its low solubility in the physiological media, chemical or enzymatic degradation, toxic effects due to its indiscriminate biodistribution, and the presence, of course, of many physiological barriers. The use of colloidal carriers for drug administration could give several advantages, such as the prolongation of circulation times into the bloodstream of drugs, and with the possibility to accumulate into required organ or tissues, and control delivery of loaded drugs. The chemical physical property, of course, of colloidal carriers, of course, such as size and surface charge and hydrophilicity, will determine the amount and kind of adsorbed protein from the bloodstream. This fact will differentiate the in vivo fate and organ biodistribution of drug-loaded systems. In the first part of this presentation, we will describe the preparation and characterization of lipid nanoparticles containing riluzole and the evaluation by in vivo experiments of their suitability as brain drug delivery systems. It doesn't work. Briefly, solid lipid nanoparticles are colloidal nanostructures made from physiological and well-tolerated ingredients which possess several advantages, such as biocompatibility and feasibility to scaling up, that is very important. Moreover, these particles have many potential therapeutical applications, such as drug delivery system for neuroactive drugs. In fact, due to their lipophilic feature as well as a small size, this particle could cross the blood-brain barrier by mechanisms as transcytosis, endocytosis, or modulation of tight junction. So we have prepared lipid nanoparticles containing riluzole. Riluzole is a potent neuroactive drug used for the treatment of amyotrophic lateral sclerosis. We have obtained nanoparticles by using the warm oil in water microemulsion technique, and obtained particles present a mean size lower than 100 nanometers and a very high drug loading equal to 40.5 weight%. Yes. In vivo experiments were carried out by using adult rats, which were previously immunized by using the experimental allergic encephalomyelitis. For carrying out this experiment, two formulations were used: the free drug, that is an aqueous suspension of riluzole, and riluzole loaded into lipid nanoparticles. Both formulations were administered by the intraperitoneal route and as a pre-treatment, the 7th day after immunization. It was found that rats treated with lipid nanoparticles containing riluzole developed clinical signs of experimental allergic encephalomyelitis later than rats treated with free drug. Moreover, after 8, 16 hours and 30 hours post-injection, the amount of riluzole into the brain, serum, liver, spleen, kidney, and lungs were determined, and it was found that the concentration of riluzole achieved into the brain was higher than when it is administered as riluzole-loaded lipid nanoparticles compared to free riluzole. Moreover, riluzole accumulates into no-target organs to a much lower extent than free drug. In this graphic, the level of riluzole into the brain tissue after administration of free or riluzole-loaded lipid nanoparticle is better evidenced. So in conclusion, we have prepared lipid nanoparticles containing riluzole, which showed colloidal size, very low size, and high drug loading, a greater efficacy than free riluzole in rats, a higher capability to carry the drug into the brain, and a lower indiscriminate biodistribution, so probably lower toxic effects. In the second part of this presentation, we will describe the synthesis and characterization of two novel amphiphilic graft copolymers based on alpha-beta poly and 2-hydroxyethyl diel-aspartamide, that is a PHEA polymer. From these copolymers, we have obtained polymeric micelles potentially useful as brain-targeted drug delivery systems. The targeting moiety chosen was twin-80 because from the literature we know that colloidal carriers bearing PS onto the surface easily cross the blood-brain barrier by a receptor-mediated mechanism after the adsorption from the bloodstream of ApoE, that is by mimicking the low-density lipoprotein mechanism. Briefly, polymeric micelles are core shell nanostructures with a mean size ranging between 5 and 100 nanometers, which are formed from the self-assembling in aqueous media of amphiphilic copolymers. So we have chosen for this work PHEA as a hydrophilic portion to obtain an amphiphilic copolymers. PHEA is a synthetic and biocompatible polymer with high solubility in water, which present an high feasibility to derivatization thanks to the presence of many hydroxylic groups onto the backbone. It was chosen for grafting of hydrophobic portion, and in particular we have chosen dialpolylactic acid, PLA, to obtain amphiphilic copolymers. PLA is a synthetic, biocompatible and biodegradable polymer. Very interesting. So this work is based on many steps, from the synthesis of two copolymers with different reaction, and to the preparation of micelles, polymeric micelles, and their characterization from the chemical, physical to biological point of view. Firstly, PHEA was functionalized with ethylenediamine to obtain PHEA EDA copolymer by following a synthetic strategy developed in the past in our lab. After this, PHEA EDA was functionalized with PS to obtain PHEA EDA PS copolymer. This reaction was carried out in organic environment and by using paranitrophenyl carbonate as activating agent for PS alcoholic groups. The proper amount of PS and PMPC were added as indicated in this slide. After purification, the PHEA EDA PS copolymer was properly characterized by using two techniques. The proton NMR and FTR analysis, which confirmed that the derivatization reaction was carried out. In particular, the degree of derivatization in PS, determined by the proton NMR spectrum, resulted to be equal to 1.2 mol%. In order to obtain an amphiphilic copolymer, PHA-EDA-PS copolymer was functionalized with PLA to obtain a copolymer with a very long name that we call, starting from now, PS-targeted copolymer. This reaction was carried out by using a N-hydroxysuccinimidic derivative of PLA and by using diethylamine as catalyst. And the proper amount of PLA, NHS, and diethylamine were added according as reported in this slide. Also, PHA-EDA copolymer was functionalized with PLA to obtain a copolymer that we call not targeted copolymer. After preparation, both PS-targeted and not targeted copolymers were properly characterized by using proton NMR and FTR analysis, which confirmed the occurrence of derivatization reaction. In particular, the degree of derivatization in PLA, determined by using the proton NMR spectrum, resulted to be equal to 0.54 and 0.60 mole %, respectively for PS-targeted and not targeted copolymers. The weight average molecular weights values determined in organic environment and by using side exclusion chromatography resulted to be in accordance with the theoretical ones. As both PS-targeted and not-targeted copolymers are amphiphilic, the dialysis method was chosen to obtain polymeric micelles in aqueous medium, and after preparation, micelles were characterized. The critical aggregation concentration values were determined by using fluorescence spectroscopy and Pyren as a fluorescence probe. It was found that for both PS-targeted and not-targeted copolymers and micelles, the CSC values were in the order of micromolar, and this value was lower for PS-targeted copolymer. Moreover, mean size, PDI, and zeta potential values were determined by using photocorrelation spectroscopy, and it was found that mean size were in about a dozen of nanometers for both kind of micelles, having PS no influence on a little influence on the micelle size. Moreover, zeta potential values resulted to be positive, as expected from the chemical composition of starting copolymer, that is PHA-EDA. In order to determine the chemical composition of the surface layer of both not targeted and PS-targeted micelles, X-ray photoelectron spectroscopy was used, and in particular in this table we report the binding energies of the main XPS peaks of both and the chemical composition of both not targeted and PS-targeted micelle surface. A curve-fitting procedure was used to determine the relative amount of carbon, nitrogen, and oxygen spaces on the micelle surface. And the main difference between both surfaces was found in the carbon atoms detected as CO spaces that was higher on the PS-targeted micelle surface. In the literature, it is reported that this bound is this atom, this peak, is an indicator of the presence of polyethylene glycol portion of PS moiety. In order to determine the possibility to administer these micelles for several administration routes, an in vitro cell viability test was carried out by using different cell lines and also different concentration of both PS-targeted and not-targeted copolymers below and above the critical aggregation concentration, and it was found that cell viability values were very high, higher than 80%, so demonstrating the absence of toxicity of both kind of micelles. Finally, in order to determine the possibility to circulate into the bloodstream escaping from macrophagic uptake, an in vitro cell uptake test was carried out by using macrophagic cells. We use also neuroblastoma cells for carrying out this experiment. And we loaded both kind of micelles with rhodamine, that is a fluorescence probe. After 4 hours of incubation with fluorescence micelles, it was found that neuroblastoma cells were able to internalize both kind of micelles, as can be seen from this red spot inside of cells, while macrophagic cells were able to internalize a higher amount of not targeted micelles compared to PS-targeted ones, demonstrating a significant difference in the surface properties of PS-targeted micelles compared to not targeted ones. So in conclusion, we have synthesized 2 novel graft amphiphilic copolymers that we call PS-targeted and not-targeted copolymers that were able to self-aggregate in aqueous media to form polymeric micelles, which possess very small size, about a dozen of nanometers, and not present cytotoxic effect on cells. And in particular, the PS-targeted micelles were able to escape from the macrophagic uptake, and so for us are potentially useful for drug targeting to the brain. Thank you to everybody and to my group, research group that is in Palermo.
Thank you, Dr. Craparo. Now the session is open for questions. Is there a question? Yeah.
Thank you. Only a curiosity, did you try something else like paranitrophenyl carbonate as, for example, carbonyl dimidazole, in order to derivatize your polymer?
No, because we start with paranitrophenyl carbonate and it was okay. The reaction, the condition were okay. It was the first activating agent. So with other reaction we had some problems. So in this case it was okay. We use a very low amount of activating agent, so we don't have cross reticulation. No, it's really a very small amount, because we use an amount relating to a very small amount compared to a mini group on the polymer, starting polymer. So a really small amount.
Okay, you use this reaction in a very short time. I mean, the first step is very short, and then you put the amino group.
What? Sorry,
the first part of reaction, the activation with paranitrophenyl carbonate, is performed in a very short time, at 3 hours.
Yes.
Okay. Thank you very much.
In 10 degrees. You're welcome.
I have a curiosity on the animal model that you use for solid lipid nanoparticles, because I'm not an expert. Animal model, because I know, but I'm not an expert of animal models, that for this pathology, amyotrophic lateral sclerosis, the only model is SOD deficient mice. So why you choose this model? You know some?
I'm not an expert, but the group researcher, what? No, people who want to try to do this experiment. I'm sorry. The group of physiologists that want to try this drug ask us to prepare lipid nanoparticles containing riduzole, so they choose this model. I think they have only this, I don't know, really.
Thank you.
Thank you.
So, you derivatize your polyhydroxyethylaspartamide with ethylene diamine. Did you have any cross reaction, because this is a dysfunctional reagent?
So
how could you avoid the cross linking?
Yeah, I understood.
And I have another question. Yes. Just a technical question, because I don't use the dialysis method for preparation of micelles, and you showed that you used this method with the dialysis tubes. I usually don't use that because I think that the dimethyl sulfoxide may
remain.
Exactly. So, can you give me some advice about it, the use of this?
So regarding the reaction of derivatization of PHEA, we are sure that we haven't a cross-linking reaction because we operate in a very dilute solution, and we also the addiction of reactant to the polymer the activating PHEA was added to ethylenediamine, so we operate in a particular way in order to avoid this reaction. And of course, we determine the yield, yeah. And we usually analyze the polymer by side exclusion chromatography in order to be sure to have higher spaces with higher molecular weight, not only cross-linking, but also higher, only, for example, 2 or 3 polymer chains together. So we are sure that in the chosen derivatization reaction, we haven't cross-linking reaction. So regarding the dialysis method, we usually try to operate in different way. We can use also another method that is, but maybe it's better for incorporating drugs. The other method that we usually use. This is only a way to favorite the core shell formation from the copolymer. I try in other way, but this was the best way to obtain colloidal nanostructures. I mean, by dispersing the polymer in DMSO, and then we carried out using the dialysis. Is the best way in this case for this copolymer to obtain polymeric micelles with this very small size, because if we change the method, we could have quite bigger structure. So it's better for having core shell nanostructure with this, of course, with these copolymers.
So thank you for the answer, and just the last short question. Yes. Do you use some special DMSO resistant membranes for doing that?
Yes, we use, of course, the volume of DMSO is quite small compared to the volume of water. We put the copolymer solution, organic solution, in water, and then we add this solution to the dialysis membrane that is from Spectrapore.
Ah, okay.
Yes, of course, are resistant to the degradation due to the organic solvent. Yes.
So, do you have an idea to explain how your micelles can escape from macrophagic uptake?
I think first that the shell of these micelles is hydrophobic, firstly, quite hydrophilic. And also we have the twin AT that possess a short portion of polyethylene glycol moiety. So I think that the significant difference between this kind of micelles is the presence of the surfactant that possess a polyethylene glycol portion. I don't know if it will be necessary to add some polyethylene glycol moieties with higher molecular weight to obtain stealth properties. We have to try this. We have also a derivative of PHA because it's very easy to derivatize this polymer. We have a pegylated polyaspartamide derivative. So we have to try, we are trying to obtain also this kind of copolymer. But of course, the circulation and the reaction with the potential interaction with the receptor will happen in a very short time. We incubate our copolymer for 4 hours with macrophagic cells. So I think it's a very long time, no? Okay.
Thank you again, Dr. Craparo.
Welcome.